Okay, so now we're going to go ahead and start lecture 6. And this one is going to start talking about how a nerve impulse actually works. We gave you some basic ideas, or I gave you some basic ideas of how what a nerve looks like, how the basic nervous system is set up, and I also talked about the unsung heroes, the glial cells. Now we're going to move on to talking about how a nerve impulse works. And so the thing that I kind of alluded to earlier, and you've seen this even when I talked about muscle membranes, is something about positive charges and how the outside at rest for a nerve or muscle cell is more positive than the inside of that muscle cell or nerve. And I'm talking about across the cell membrane. Hopefully you remember from your introductory classes or your cell biology class that a cell membrane is made up of phospholipids. Those phospholipids are hydrophobic, and they do have a hydrophilic head, but they're hydrophobic internally. And so a lot of things aren't very permeable to that cell membrane unless they go through a protein channel. So again, what I'm hoping is you have some familiarity with the cell membrane. I'll help add to that and remind you a few things. But remember that for the most part, a cell membrane is made up of this phospholipid bilayer, has different proteins associated with it and carbohydrates, receptors, and all sorts of things, along with what we call protein channels that can open and close. These channels are like doors that can allow some things to enter and some things to exit the cell. And so this is what allows for a voltage-gated difference to occur on the outside of the cell versus the inside of the cell. And this voltage differences in electrical charge is due to the type of ions that are outside and inside. And again, where would the ions come from? This is coming from different atoms that have a slightly different electrical charge based on whether or not they have more protons to electrons. If they have more protons in regards to electrons, those atoms have what we call a positive charge or a cation. And one of the things when you think about cat with the letter T in the middle, it has a positive, because a T looks like a positive sign. So this is one way to remember that some atoms, like sodium for instance, are cations, while other atoms like chlorine is negatively charged. There are more electrons than there are protons in the nucleus. So the electrons, as you remember, go through or outside the cell, the shells and things like that. So those would be called anions. So anions have a negative charge. And it's the relative abundance of positive charge and negative charge that makes this difference. And then again, so when we're looking at the notes, you can see plasma membrane is another term that we can pretty much interchangeably use with cell membrane. It's going to be made up of those phospholipids. And so the main players that involved, as I kind of alluded to, is sodium, which is a cation, chlorine, which is an anion, potassium, which is a positively charged cation. Calcium is also positively charged and involved quite a bit. And even though it's not listed here, negatively charged proteins, because proteins that are found inside the cell, there's quite a few of them, obviously, lots. And those amino acids, some of them can be negatively charged. And so that can all kind of cumulatively make the inside more negative than the outside. So the negatively charged proteins on the inside of the cell, the chloride atoms, and then the relative abundance of potassium and sodium inside and outside give a net charge of being more negative on the inside of the cell than on the outside during rest. And we call this the resting membrane potential. So neurons and muscles have this negatively charged resting membrane potential. And it'll make more sense when I show you the videos, but basically during a nerve impulse, we have a rapid reversal where momentarily a portion of that axon or nerve will be positively charged because sodium will enter into the cell rapidly for a moment. So this is representing some of what you would see on a cell membrane. So we have, let me just kind of orientate you. Hopefully this is easy to look at and know what's going on. Here we have a cell membrane, and you can see that this is the phospholipid bilayer. Again, I'm hoping that that rings a bell from your intro classes. And so if you look at the cell membrane, you can see the little blue circles are part of this phospholipid that we call hydrophilic. And then coming off of it are carbon chains of lipids. And if you're familiar with fats, try to mix up fats and water, they don't mix. So that would be the hydrophobic portion, the hate water. So the blue part is the hydrophilic heads. It's going to be made up of various atoms. I had to go look at a phospholipid, built phospho. So it's going to be phosphates, for instance. And so they're going to be hydrophilic facing towards the water. So so there's two layers of it. There's the one layer that you can see on the outside if you look at my cursor. And again, I'm hoping this is just super easy reminder. And so here's our hydrophilic heads, hydrophobic tails. Again, it's going to be carbon atoms. It's fats. And then here's another layer and they face towards the water being on the outside and the inside. And if you remember, the watery stuff inside a cell is called a cytosol. And so this is the extracellular fluids, and these yellow dots represent sodium atoms. And so you can see, relatively speaking, there's a lot of sodium atoms on the outside in comparison to the inside, helping make the outside more positive. Now you'll notice there's actually quite a few potassiums on the inside, but it's the relative amounts. And keep in mind, there's also chlorine atoms on the inside of the cell, along with negatively charged proteins. So it's the overall net effect that makes the inside more negative than the outside during rest. Now, what actually maintains this difference is a sodium-potassium pump. And so what you'll notice is this is a picture showing you kind of a before and after. So basically the channel's opening and closing, and I'll show you some animations, obviously, at some point with a YouTube picture animation. But what you're seeing in this picture on the left is that the sodium channel opens and 3 sodium atoms enter into the inside of the cell. Then, or excuse me, I'll take that back. Let me reverse that. Here you see 3 sodium atoms are being put into these receptors. So let me just make sure you understand what the sodium potassium pump is doing. The sodium potassium pump is actually pumping the sodium out. This is what maintains this difference in charge. So what happens in this picture here is you see this here is the channel protein. These 3 sodiums are coming in and attaching. So there's 2 already attached in yellow. And then here's a third space where this sodium atom is going to jump in and fit. At the same time, these 3 sodiums are coming into the channel, 2 potassiums are leaving. And then it opens to the outside, and then the sodium is pumped out, and then whatever potassium's out here flows in. So we have 3 sodiums leaving and 2 potassiums coming in. So 3 positives are leaving, 2 positives are coming in. Because remember, positive potassium is positively charged. So 2 positively charged atoms are coming into the cell, 3 positive sodiums are leaving every time this channel works. And what do you think is probably helping pump and move this channel? Well, it would be andesine triphosphate, the fuel source of cells. And so the sodium is being pumped out, forming a concentration gradient where you have more sodium on the outside than sodium on the inside. Because what do we know about diffusion? We move from an area of higher concentration to an area of lower concentration. So when you're going against that concentration gradient because you're pushing so much sodium out, it actually takes energy to push more sodium out. And where do you get that energy? Again, it's going to be anderson triphosphate, ATP. And so the outside becomes more positive because of all the sodium going out. Now you might be thinking, well, what about the potassium? You're bringing 2 in, but again, it's relatively speaking, you're putting out 3 positives to every 2. So that automatically, mathematically makes a difference. There's also negatively charged proteins on the inside of the cell along with chloride. Not only that, the potassium that you pump in, some of it leaks out. And so that's what's being shown in this picture on the right. This is a potassium channel. And so it's usually slightly open. That's not necessarily what it looks like in this picture. I realize it looks like it's wide open. But during the resting membrane potential, the potassium channel is just a little bit open and a little bit of potassium's leaving, making it a leaky channel. So it'd be kind of like you were in a boat full of water because you sprung a leak or whatever. So you start bailing water out. And so you're bailing out the potassium or bailing out the water. But a little bit of potassium keeps coming back in. So here we are pumping the potassium into the cell on the left side, and some of it is leaking out during the resting membrane potential. That obviously means there's a point where it's not resting and the potassium channels are going to open up wide, and we'll get to that momentarily. Right now, I just want you to get the gist of what a resting membrane potential is doing. The sodium potassium pump is pumping more sodium out, 3 sodiums out, a couple of potassiums are coming in. It's using anderson triphosphate as the fuel source. And the little bit of potassium that was pumped in, some of it is leaking out. And then the net overall charge is more negative on the inside of the cell and more positive on the outside. Let me see if I can provide a little animation to help explain that further. Okay, so now I'm going to show you an animation of the sodium potassium pump. You can see the cell membrane. Now this, or actually we call this the plasma membrane. You know, the cell membrane would include the proteins and everything else that's associated with it. These are those phospholipids. There's one layer here, and then the dividing lines right here, and another layer down here. So this is the hydrophilic heads. Hydrophilic heads like water. Hydrophobic tails are fats and hate water, and so the water on the outside and water on the inside will have the ions. And again, the channels will be involved in moving, or the transports or pumps will be involved in moving these ions. So we're going to first tell you about one called active transport. This is the one that requires ATP. We also have facilitated transport, if you remember that from your cell biology. That active transport allows sodium or potassium to be pushed past its concentration gradient or its electrical chemical gradient.
Transport that requires the input of energy in the form of ATP.
So
the proteins that conduct this form of transport are often called pumps because they force molecules or ions to move from an area of low concentration to an area of high concentration. This is commonly referred to as against the concentration gradient.
Yep. So remember, the concentration gradient is the sodium would normally diffuse into the cell because the sodium is in a higher concentration on the outside of the cell, while the potassium is in a higher concentration on the inside. So it takes energy to push the sodium out, and it takes energy to move potassium back into the cell because it's going against the concentration gradient of those particular cations. Now, yeah, so that's the main thing. So,
gradient. One of the more common examples of active transport is the sodium-potassium pump. The job of the sodium-potassium pump is to move sodium ions, or Na+, out of the cell, and potassium ions, or K+, into the cell. Let's take a closer look at how this happens. The sodium-potassium pump has binding sites for 3 sodium and 2 potassium ions. Let's follow the movement of the sodium ions first. After 3 sodium ions are positioned within the carrier protein, an ATP molecule is split.
So here you can see that these are special receptors. Again, remember this is, if you remember a little bit of biochemistry, this is a protein that's going to have a special groove that can hold on to the sodium. It'll be, if you were really thinking about the molecule level, it'll be something that maybe has kind of the right shape and maybe the electrical charge. I'm not sure exactly what that region would look like, but it's going to be something like that at the molecular level. And then here we have, so this is all protein, and then this andesite triphosphate will come along and attach. Now remember, this is going in again with your basic chemistry like covalent bonds and stuff like that with the phosphate atoms. And so when you break a covalent bond, you're providing energy. And that energy in this case actually changes the shape of the protein, because this is a protein. And with the extra bonds and removable bonds, the shape of the protein will shift in such a way that it will eject these sodium atoms. And then the potassium will be receptive for picking up potassium.
Releasing phosphate. This phosphate binds to a location on the...
So when the ATP broke off that phosphate, obviously the andesite diphosphate left, for di for meaning 2. And this is what changes the conformation of the protein.
The exterior of the carrier protein, causing the protein to change shape. As the protein changes shape, the 3 sodium ions are released to the other side of the membrane. Next, 2 potassium ions position themselves within the carrier protein, causing it to undergo another change in shape. In the process, the phosphate molecule is released. Once the phosphate molecule is released, the carrier protein expels the potassium ions into the interior of the cell. The carrier then resumes its initial shape, completing the cycle.
And thus more sodium can come in and hook up with the pump. Now, I want you to keep in mind that this sodium-potassium pump is always active in the background, irrespective of a nerve is depolarizing or repolarizing or so forth. This is what's maintaining the overall difference in charge. And so it's just running constantly. We haven't got into how a nerve impulse works, but we're at the point of describing what is setting up the nerve impulse. We got to get this membrane potential set up. And this, like I said, the sodium potassium pump is working as long as you're alive and your cells are alive, it's active.
Notice that for every 3 sodium ions leaving the cell, 2 potassium ions enter. Since both sodium and potassium ions have a positive charge, this unequal movement causes an electrical gradient to develop across the plasma membrane of the cell.
So the outside is more positive than the inside. Remember, it's the relative amount of, there's a lot more sodium out here in relationship to the sodium on the inside. Now granted, the potassium is also positively charged, but some of that potassium is leaking out in a channel that's not shown in this picture. And what else do we have inside? Chlorine atoms and negatively charged proteins.
Cell. A number of cellular processes, including the generation of nerve impulses, use this electrical gradient.
Obviously, I certainly encourage for you to look at videos whenever you have a moment. There's a lot of good stuff on YouTube. Let me go back to, so I'm going to show you this animation. I have some few little issues with it that I will explain as we go through. This is going to talk about the neuron resting membrane potential. One of the big flaws of this video is it doesn't mention anything about the sodium potassium pump. The other kind of big flaws you're going to hear in a moment that probably will distract you a little bit is like a rainforest sound in this video. So, I mean, we don't have rainforest inside our head, but you're going to hear crickets and rain, whatever. But I think the animation's nice, so I wanted you to get that. And then I'll just kind of correct some parts of the video that I think need a little explanation. But the first thing we'll do is we're looking at, obviously, motor neurons. I don't know if it's supposed to be depicting the central nervous system, but remember a lot of the motor neurons is going to be, the fibers are going to go outside of the central nervous system. So you're going to have more interneurons inside the central nervous system. That said, what we have here is dendrites. This is a cell body, and then this would be the axon. And as you remember, this would be the myelin sheath. And then this would be the nodes around the veer. And so we see all sorts of neurons throughout this image.
Unstimulated, neurons maintain a constant electrical difference or potential across their cell membranes. This potential, called resting potential, is always negative inside the cell and ranges from -40 to -90 millivolts.
So we could have a probe on the inside of the cell and tell that it was more negative internally than externally. Again, it's going to be mostly due to sodium atoms predominantly on the outside. This video doesn't mention the sodium atoms. It only talks about potassium. That's an issue I have with this video.
If a neuron is stimulated, the negative potential.
So what these yellow dots are representing are what we call sodium sparks, or positively charged sparks. They're little bits of depolarization that have occurred, and that's what this is showing here. So remember, information travels down a nerve, down the dendrites, into the cell body, where and we're going to get into this later, but the overall charge forms what we call a graded potential, and I'll get into that more in future lecture, because we're probably not going to get through everything to get to that action potential yet in this particular video, or we'll just get to the beginning of it. But this positively charged signal then reaches what we call the axon hillock, and then either results in a true action potential that will travel down the neuron, a true nerve impulse that travels down the neuron. And again, that'll be sodium coming into the cell that causes this rapid reversal, and it'll make more sense as we go down this lecture. How, and no pun intended, going down the lecture, going down the axon, but anyway, the resting membrane potential is negative overall.
Inside the neuron can be made either more or less negative depending on the stimulus.
Less negative being more positive, obviously.
If potential is made sufficiently less negative, it reaches a level called threshold.
So that means, and we're going to get into this more, but this means that when it became less negative, the inside went from a -90 or 80 millivolts to up to -50 millivolts. So it goes from less negative to, from -90 millivolts or 80 millivolts up to a -50 millivolts or so. And when it does so, it's going to open up all sorts of sodium channels. That'll come down later into the lectures, either today or down in the next lecture. But that is what we call threshold. And then if it reaches threshold, we have this all or nothing event, which causes the nerve impulse to take place.
Threshold, and an action potential is triggered. During the action potential, the neuron suddenly becomes 20 to 50 millivolts positive inside. Action potentials last a few milliseconds before the cell.
So once we've reached that threshold, which I thought should be around a -30 or something like that, it then jumps up, as they mentioned, to around 30 millivolts when the rest of the sodium channels open. It's just whatever triggers those sodium channels to open, which we'll describe better in the upcoming lectures. And then, as you saw, the nerve impulse is just kind of by itself all the way down the nerve. It's not the whole nerve fiber firing at once. It's actually a little portion traveling down the nerve, a bundle of more positive charge that will just literally travel down the axon. The myelin sheath speeds up that nerve impulse, as we'll describe a little bit later. But once it reaches the axon hillock, that's where neural transmitters will be released. And I'll describe how that happens in a future lecture as well.
Restores its negative resting potential.
So after that action potential, the nerve goes back to resting.
The cell membrane of a neuron encloses cytoplasm with various ions dissolved in it. The neuron itself is immersed in a salt solution, the extracellular fluid.
So the extracellular fluid is going to be predominantly sodium. There is going to be some potassium out here as well, relatively equal to the amount of potassium on the inside. And then we have these negative organic compounds, which often are proteins from what I remember and understand. They're going to be negatively charged amino acids. So the overall net effect is negative. Now, it doesn't really talk about the sodium-potassium pump. That's where I think this...
The ions of the cytoplasm consist mainly of...
So here's our negatively charged organic compounds like proteins. And then these blue things are representing the potassium atoms. Now, there's some channels that allow the potassium to leak out, as I mentioned in the previous slide or two.
Positively charged potassium ions and large negatively charged organic molecules, such as proteins. Outside the cell, the extracellular fluid contains mostly positively charged sodium ions and negatively charged chloride ions. Since charged particles cannot pass through the lipids that make up cell membranes, they must travel through channel proteins that extend through the membrane.
Again, we have our cell plasma membrane. We've got your phospholipid bilayer. And then this is the proteins channels that will be open or closed depending on whether or not we're going through a nerve impulse. During resting membrane, when the nerve is not firing, the sodium channels are closed, while the potassium channels are slightly open and leaky. Now, what's not shown in this picture that I showed you in the previous animation and the previous slide is the sodium-potassium pump is always active in the background, pumping the 3 sodiums out, bringing 2 potassiums in. But those 2 potassiums can leak out of a potassium channel. The sodium channels are completely closed, though, during the resting membrane potential. So more sodium stays out. And while some potassium is pumped back in by the sodium-potassium pump, some of that potassium leaks back out into a relative equilibrium of potassium part. The potassium is relatively equal. But the overall charge is still more positive on the outside because there's a lot more sodium atoms. In this video, they mentioned some chloride atoms on the outside, but overall, again, there's a lot more negatively charged anions on the inside of the cell.
Brain. In an unstimulated neuron, only potassium ions can cross the...
So this picture is showing you what, in a gross scale, what obviously you would be able to see the channels like this, but this is supposed to represent the sodium channels closed, and this is supposed to represent the potassium channels slightly open and leaky, allowing some of the potassium to move out. What is not shown in this animation is the sodium-potassium pump that is still actively engaged.
The membrane, traveling through specific proteins called potassium channels. Although sodium channels are also present, in unstimulated neurons, they remain closed. Since only potassium ions can cross the membrane, and potassium ions are more concentrated inside the cell, they diffuse.
I mean, they're initially more concentrated on the inside of the cell because the sodium-potassium pump pumped the potassium in, and then it leaks out of the channels.
out of the cell, leaving the large negatively charged organic ions behind.
But again, remember, there's sodium out here. It's not just the potassium, it's the sodium, potassium. Overall is out here is higher and more positive than the inside, giving you that more negatively charged molecules on the inside of the cell when the nerve is not firing meaning not going under a nerve impulse.
As more and more positively charged potassium ions leave, the inside of the cell becomes increasingly negative. But since opposite charges attract, as potassium ions diffuse out, an electrical force develops that tends to pull them back inside.
So what it really means is there's some diffusion going back and forth in regards to the charges across the membrane, what channels are open, and again, the sodium potassium pump is also pumping some of this potassium back in, which isn't really being described here.
At some point, the diffusion of potassium ions out of the neuron due to concentration differences is balanced by the electrical attraction tending to pull them back inside. This is
electrical, remember, because these are more positive than the negative inside. There's at least an attraction for the positively charged ions to go towards the negatively charged organic molecules. That's what he's talking about. But it never reaches an equilibrium. The inside still remains more negative than the outside.
Is the point at which neurons reach resting potential. Reaching resting potential in this way does not require significant changes in the potassium concentration inside or outside the cell. Only about 1/10,000th of the potassium ions initially inside a neuron must leave to...
I don't know if you caught that, but what he's saying is it only takes a few molecules to maintain this kind of difference in charge. These are all millivolts. It doesn't take many cations and ions, anions, and movement to make a big change in the millivolts. So it's not a huge amount necessarily coming and going to get these changes in charge. That's the one thing that you should understand. It's not a huge amount of potassium entering, exiting the cell, or sodium. It's all done relatively a few atoms and so forth. And that's enough to change the millivolts inside the atom, or excuse me, inside the cell.
To create a resting potential of -60 millivolts. But it is action potentials, not resting potentials, that carry information through a nervous system.
So obviously we're going to get action potentials as we continue this lecture. Let me see if there's anything else I want to mention in regards to membrane potential before we move to the next step. So I'm going to hit a couple parts of this video here just to show you, again, a little bit of animation. Here's our cell membrane. And this is just to get kind of reinforced membrane potential.
Membrane voltage refers to the difference of electric charges.
So here we have our electric or organic anions, and then we have our major players, potassium, sodium, and then chloride isn't shown in this picture.
Across a cell membrane. Most cells have a negative transmembrane potential. Because membrane potential is defined relative to the exterior of the cell, the negative sign means the cell has more negative charges on the inside. There are two basic rules governing the movement of ions. They move from higher to lower concentration.
So as you know, diffusion moves from an area of higher concentration to an area of lower concentration. But there's also electrical chemical gradient that's formed. So if the inside's more negative, the positive is going to want to move internally.
Just like any other molecules. Being charge-bearing particles, ions also move away from like charges.
So obviously positive repels for positive, negative attracts positive.
And toward opposite charges. In the case of the cell membrane, there is a third factor that controls ion movement: the permeability of the membrane to different ions. Permeability is achieved by opening or closing passageways for specific ions, called.
So these are ion channels, and you can see the sodium channel opened in this particular picture. That's what happens actually when a nerve impulse occurs.
Ion channels.
Potassium leaves.
Permeability can change when the cell adopts a different physiological state. Consider this example. Two solutions of different concentrations of sodium chloride are separated by a membrane. If the membrane is equally permeable to both sodium and chloride, both ions will diffuse from higher to lower concentration, and the two solutions will eventually have the same concentration. Note that the electric charges remain the same on both sides and membrane potential is 0. Now let's assume that the membrane is permeable only to the positively charged sodium ions, letting them flow down the concentration gradient while blocking the negatively charged chloride ions.
So you can see that the sodium is moving from an area of higher concentration to an area of lower concentration relative to sodium. And in doing so, the inside becomes more negative than the outside because there's more chlorine atoms on the inside, relatively speaking, to sodium atoms in regards to the outside, where there's more positively charged sodium. So this is, again, relative. And again, this membrane right now is supposed to be permeable only to sodium.
Ions from crossing to the other side. This would result in one solution becoming increasingly positive and the other increasingly negative. Since opposite charges attract and like charges repel, positive sodium ions are now under influence of two forces. Diffusion force drives them in one direction, while electrostatic force drives them in the opposite.
So what she's saying is that the positive sodium now has an interest in attraction and going back the opposite direction.
The equilibrium is reached when these two forces completely counteract, at which point the net movement of sodium is 0. Note that there is now a difference of electric charge across the membrane. There is also a membrane. These gradients are used to calculate their equilibrium.
Okay, so now we're going to look at the relative amount of sodium to potassium and chlorine. And so I hope this video might help clarify a little bit. Because remember, they're still not shown in this picture the negatively charged ions on the inside of the cell. But this is millimoles. I don't know if you're familiar with that from chemistry. So we have 150 millimoles of sodium on the outside of this example, 5 millimoles of potassium on the outside as well, and 120 millimoles of chlorine on the outside. So in this particular picture right now, we're seeing what I would think suggests that the outside is more negative. Let's see what happens.
Equilibrium potentials. The positive and negative signs represent the direction of membrane potential. Because sodium gradient is directed into the cell, its equilibrium potential must be positive to.
So that would be due to diffusion. There's a lot more sodium here than here. And again, this is just providing an example of how this is all set up.
Drive sodium out. Potassium has the reverse concentration gradient, hence negative equilibrium potential.
Because it's going to go from an area of higher concentration of potassium to an area of lower concentration of potassium.
Chloride has the same inward concentration direction as sodium, but because it's a negative charge, it requires a negative environment inside the cell to push it out. The resting membrane potential of a neuron is about negative 70 millivolts. Notice that only chloride has the equilibrium potential near this value. This means chloride is in equilibrium in resting neurons, while sodium and potassium are not. This is because there is an active transport to keep sodium and potassium.
When she said active transport, is the sodium-potassium pump.
Out of equilibrium.
So here's our sodium-potassium pump. It's more positive on the outside than on the inside. Here's our negatively charged organic anions.
This is carried out by the sodium-potassium pump, which constantly brings potassium in and pumps sodium.
So you have 3 sodiums left and 2 potassiums came in. Remember, ATP is utilized.
Out of the cell. The resulting resting potential, while costly to maintain, is essential to generation of action potentials when the cell is stimulated.
So we're going to get to this a little bit later, but what's the bottom line? The inside is more negative than the outside. There's a lot more sodium on the outside. And this is what's going to lead to the action potential. Resting membrane potential, -70 to -90 millivolts. The sodium potassium pump is maintaining that for the most part. And a little bit of potassium is leaking. There is obviously some chlorine atoms on the outside, but again, there's still a lot more negatively charged anions on the inside. Now, that may have been a little bit, quite a few examples, but I'm hoping that it helps you to visualize and have a better appreciation. So here again is our sodium potassium pump activated. Sodium is in yellow being pumped out. Potassium is being pumped in. Excuse me. Sodium is being pumped out. Potassium is being pumped in, so that's correct. And then we have a little bit of a leaky potassium pump that's allowing for some equilibrium of potassium to occur. And then if we stick our needles in, you can see that overall still the inside of the axon is negative in comparison to the outside. And we call that a resting membrane potential. And every textbook and examples, it seems like we're seeing -60 millivolts or -80 millivolts, but it kind of goes into this range. And so here again is another picture showing you the resting membrane potential and how we have negatively charged proteins. We have some chlorine, but we also have some chloride out here as well. But again, it's mostly this net effect of more sodium on the outside that is making, relative to the inside, that's making it more negative because the sodium channels are closed. And again, the sodium potassium pump is always working on it and helping to maintain this. So now we're going to start describing the nerve impulse. Again, this is a sudden and rapid reversal of charge, but only within a region where that one little region becomes more positively charged. What essentially happens is that sodium channels open briefly for a split second or so, milliseconds, 1 to 2 milliseconds, opens up briefly, allows a few sodium cations to enter and then closes. That momentarily makes that little region of the long neuron more positive. And then, as you've seen in the previous animation where we have the rainforest neuron, that positively charged diffusion travels down the neuron. And I haven't really described how it continues, but essentially what happens is there's sodium channels up and down this axon that will open. And this will make more sense when I show you more pictures. Nerve impulses are very rapid, and we call these action potentials. So so again, what is happening in action potential? This is a sodium channel, and I'll get into more details about the sodium channel. But the sodium channel briefly opens, sodium atoms come in, and then it closes again, like this. And then the inside does what we call depolarization, meaning that the inside becomes less negative. In this case, it's showing going from -60 millivolts up to a -50 millivolts. So it becomes less negative or more positive, if you want to. But either way, just depending on how you look at it, it's becoming less negative. And again, this is very rapid, 1 to 2 milliseconds. These channels are what we call voltage-gated channels. And I'll get into more detail on how these channels actually open and close relative to their charge. But basically, they are turned on by positive charge. So I've thrown quite a few different ideas together, just try to bear with it, and then that'll get you to the point of understanding an action potential. Now, after this action potential has occurred down a nerve, and again, it'll make more sense as you see the whole pieces come together, we go back to our resting membrane potential. And how we do that is that we open up potassium channels from being leaky to wide open, allowing potassium to leave and bringing the inside of the nerve, making the inside of the nerve more negative again. Why? Because the positive the positive potassiums are leaving relatively rapidly, making the inside more negative. In addition, chlorine channels will open and more chlorine will come in and make the inside more negative. So it actually goes into what we call hyperpolarization, where we go from a resting membrane potential of around negative 60 millivolts, become more negative briefly, and we call that hyperpolarization, and we have a negative 70 millivolts. Now, how did it become more negative? Again, positive potassium is leaving the cell relatively rapidly, and chlorine's coming in. And together that makes the inside more negative. But it's really the sodium-potassium channels that do most of this interplay in changing the charges inside the nerve. Again, a few more pictures and slides down in this lecture, it'll make a little bit more sense. So just try to appreciate resting membrane potential. The inside is negative, around a negative 60, 70, 80 millivolts. We have depolarization. The inside is briefly more positive by sodium channels opening and allowing sodium to enter into the cell. Those sodium channels close, potassium channels open more forcefully, allowing quite a bit of potassium to leave, and then the inside becomes more negative again. And that's kind of the cycle of a nerve impulse. But again, it'll be easier to visualize when you see how it operates across a nerve and an axon. So again, depolarization, the inside becomes more positive. Hyperpolarization, the inside becomes more negative. So take some time to read these notes, and I'm going to go show you another picture and describe the action potential a little bit better. First, let's talk about the sodium channel. Now remember, the sodium channel is what we call voltage gated. That means, and if you know what voltage is, it's electrical charge, right? So this channel, and again, it channels the protein tube that allows sodium to flow into the nerve, is affected by the electrical charge around it. So in this picture here, this is a slide that you should really study. And this is going to be probably one of the slides I'd like for you to draw for your, this is one of the slides I want you to draw for this, for credit for this particular lecture. What you see here is a needle, and it's measuring the inside of, or right around the inside of this nerve. And this is the outside. It's called this is the extracellular fluid. That's what ECF stands for. ICF is the intracellular fluid. And that would be where the cytosol is, because it's the inside of the cell. Now remember, at rest, that's what this is saying. Follow this comic book strip. At rest, the membrane potential, the outside is more positive than the inside. So we know the inside is more negative. Now, the interesting thing about this channel is there's two gates to it. And what do we know? What is a gate? Well, if you have a fence and you have a gate, you know the gate opens and closes. If you don't want somebody into your yard, you close the gate and sometimes you lock it. And so we have two gates though. And that, if you ever been to, a cattle barn, they have two gates. So, or you've been to the zoo and you got the parakeets. They don't want the parakeets flying out, so they have two gates. So you can open, walk into one gate, close it, open up another gate and walk on in, out or in, depending on the direction that the tourists are moving to see the flowers or the butterfly garden or whatever. You've seen these two gates before. Or you might see in restaurants where they have two doors. And so we have an activation gate, the name of one gate, and an inactivation gate is the name of the other gate. Well, the activation gate is turned on based on the charge, and so the inactivation gate is actually used to turn it off. When I say turned on and turn off, I mean the activation gate, when it opens, allows sodium to come in. When it's closed, sodium can't come in. And the inactivation gate helps in backing up the activation gate temporarily. So there's two channels, or there's one channel and two gates. So the activation gate is closed when the cell is undergoing resting membrane potential around -70 millivolts. Notice that the inactivation gate is actually open. So there's only one gate that's closed and the other gate is actually open. But nothing is flowing through because the activation gate is closed. So the sodium is not entering. Now, and we haven't got into how this happens yet, but let's say there's an event that activates a nerve impulse. And we talked a little bit about it. The inside becomes more positive through a graded potential. And we'll get into that a little bit more in following lectures, but the inside becomes more positive. When the inside becomes more positive, around -55 millivolts, we reach what we call a threshold. That threshold opens up the activation gate. So that -55 millivolts causes the activation gate to open. And when it opens, sodium flows through. So that means something downstream from the dendrites in the cell body turned on these sodium channels. Turning it on means it brought the resting membrane potential from -70 millivolts to -55 millivolts. The activation gate opens. And when the activation gate opens, more sodium flows in. And what happens when more sodium flows in? The inside becomes more positively charged briefly in that one area. That's that depolarization. That is that rapid reversal in charge. So we move from a -70 millivolts, from a -55 millivolts, up to a +30 millivolts. So here's the sodium coming in the diagram. You can see C, if you look at my pointer, the activation gate's open, sodium is flowing in. The inside is becoming more positively charged. Then, once we reach this threshold of 30 millivolts, the inactivation gate is actually triggered to close. So it knows there's too many people coming in, COVID, so they're closing the gate. I don't want to put a COVID spin on it. We reach maximum capacity for this room, so we're going to close one of the doors. In this case, the door is the inactivation gate. And what stimulated it in this real world example? This real example is that the inside became 30 millivolts. So if you look at D on the right, and you see my cursor or pointer, the inactivation closed around 30 millivolts. That inactivation gate closes and no more sodium can enter. Now, what happens when there's no more sodium entering? The inside begins to become more negative again. And this is all happening within milliseconds. And the cool thing is this is happening in all animals with the nervous system. Sponges don't have nervous systems, but all sorts of other animals do. this is basically how it works in all animals. And so the inside starts becoming more negative again. Why is it more negative on the inside? Because there's no more positive sodium coming in. And we know that there's a lot of negatively charged proteins and so forth. What else is making it more negative that's not shown in this picture that is critical for you to realize? Potassium channels have opened and potassium is leaving. That is the most important part of what makes this more negative. It's not shown in this picture here. It'll make more sense in other slides. But what you're seeing here is that as this is dipping down, negatively charged potassium channels are open. Potassium is obviously positive. It's leaving the cell, making the inside of the cell more negative. That is called repolarization, and actually leads to what we call hyperpolarization. So what I just mentioned before, depolarization, the inside becomes more positive. Repolarization becomes more negative. Hyperpolarization becomes even more negative, and then it goes back to its resting membrane potential. Again, it's due to the movement of the sodium atoms. Sodium atoms coming in depolarizes. Potassium atoms leaving repolarizes and even causes hyperpolarization. The inactivation gate and the activation gate are directly influenced by these voltages. So if you go back to A, there were some other channels that were allowing sodium to enter. Once it reached the negative 55 millivolts, this activation gate was opened. Once it reached positive 30 millivolts, the inactivation gate comes in, closes, does not allow any more sodium to enter. The inside of the cell becomes more negative. And again, it's not shown in this picture, but it's because potassium is leaving. And once we get past that threshold of negative 55 millivolts, the activation gate closes and the inactivation gate goes back to where it was at the resting membrane. And then we're ready to go again. So if you look at E, you can see that the activation gate's coming in and closing. So no more sodium comes in, but the inactivation gate goes back to its open state. Now what's causing these gates to open and close? Again, it's the voltage, and that's changing the shape of the protein. If you want to keep in the background the idea about proteins can change their shape, and this channel changed its shape based on the voltage that was going through it. One being an activation gate, one being an inactivation gate. So again, I'm hoping, even if this seems a little complex, well, it'll take a little time and work to get through it. But think about all the smart researchers that devoted their lives to figuring this stuff out. It's really incredible. PhDs, right? I mean, sure, there's an MDs involved with science research, definitely. But this is heavy physiology work research. So people that devoted their lives to science to figure this kind of stuff out. And the cool thing is that this basic understanding is true across all the animals for all practical purposes. I'm sure there's some differences. And then again, that's the beauty of evolution, is the fact that all animals are related to everything. All life is related to everything on Earth. And so there's these common patterns we can continue to see. And where did this might have evolved from ultimately, these channels? Evolving in an ocean environment where you had salt water that was in higher concentration to the outside of the organism. So you had to regulate that salt. And they did so through things like these channels. Remember, a scientist is like a detective. You're just piecing together what you can, trying to make sense of it. Some of it will be experimental, some of it will be comparative. Obviously, nobody was there when life, the spark of life evolved So we're like a detective at the crime scene trying to put the pieces together, trying to figure out what happened. And as a physiologist, you're trying to figure out how it happens. I'm going to skip a couple slides and come back to it, but I want you to see this overarching slide here. This is also a very important slide. And then I'll go back and maybe take a look at those slides. But what we have here is what's happening in a neuron. And here's the different channels involved. I'm hoping this helps piece together some of this to you. Now remember, a nerve has got a really long axon. So this is going to be happening up and down this axon, or actually just down the axon. Information typically just flows, almost always flows just straight down the axon. And so what do we see here in this first box? And this is another one I think you really need to study and draw. What do we see in this first box? We see blue being potassium channels. We got one that is open and leaky during resting membrane potential. One that's completely closed. It might be a little bit leaky, but it's closed in this picture. And then we have two sodium channels that are closed. Notice, if you look at my cursor, that these are the activation gates, and they are closed. And if you go look at the picture, number one, it says threshold, resting membrane potential. And in this particular example, it's showing negative 60 millivolts. In other examples, we see negative 70, negative 80. Then something's triggered an action potential. And what happens is the sodium channels begin to open. The activation gate is moved out of the way. Sodium begins to flow in, and we reach a negative, in this particular example, a negative 50 millivolts. The previous slide said negative 55 millivolts. But either way, we've reached the threshold that causes the voltage gated channel, the activation gate, to open. And so the activation gate opens, sodium flows in, and then the channel that's right next to it also opens in slide number 3. So you got 1, 2, 3. So when slide 3 opens, what do we see? We see the next sodium channel opens. And this will happen all the way down the axon as the charge travels down the axon. And so the sodium channel next to it opens, and sodium begins to flow in. So the inside becomes more positive. And you can see now we're moving from, once we reach this kind of threshold, we call it an all or nothing event, and the sodium channel's open. And remember, in the previous picture, negative or positive 30 millivolts important. In this one, it shows positive 50. So there seems to be some variation across these different books that I've reviewed. But once we reach this upper threshold of 30 to 50 millivolts, the inactivation gate closes. And that's what we see in picture number 4. And so you see the bottom part, the inactivation gate is closed. No more sodium's coming in that region. And notice the potassium channel is now open. If you go from 3 to 4, the potassium channel is opening. And so what happens when the potassium channel opens? The potassium begins to flow out. And when the potassium flows out, the inside becomes more negative in the nerve. We call that repolarization. And in fact, so much of the potassium is leaving that the inside actually becomes more negative. And we call that hyperpolarization. That actually dips below the resting membrane potential. So here's our resting membrane potential around negative 60 millivolts, and we'll actually dip down to around negative 70 almost in this particular example. After that, the potassium channels close. If you go to the slide 5, and the resting membrane potential returns to around negative 60 millivolts. Again, it depends on the book and the scientists and the articles or whatever. But notice now that the sodium channels, the activation gate has closed. And the inactivation gate is now back to open. And so we're now back to our resting membrane potential. Now, if you're trying to piece this together, realize these yellow are the sodium channels that we saw here. So this is the yellow sodium channels, showing you more directly how it works. So this is showing you the same thing here, but now it's showing you the potassium channels in the background and also describing each portion where we have depolarization, repolarization, and hyperpolarization. I'll get back to these in a moment or in a little bit, but let me just briefly talk about it. So here it says depolarization, regulating the action potential. That's what AP is. Depolarization comes along, meaning that depolarization makes the inside more positive. It opens up the sodium activation channels, as I've already showed you in those different figures. The activation gate opens, sodium comes in. This is a positive feedback cycle. So that means more sodium channels down the road are going to depolarize and open. And this will continue traveling down the axon. So that's what this is showing you. At the same time, potassium channels open and potassium leaves. And when the potassium leaves, the inside becomes more negative, just like I showed you in the slide over here. That causes the inactivation gate to come in and stop the sodium channels from working. And then that's when we have repolarization. So that's what this figure or slide is about. It's kind of showing you the cycle and pattern without actually showing you the channels. And that's what's also shown in this picture. Is kind of the same thing that you saw from those two pictures, particularly the one over here, particularly the one here. This is just showing you in a graph again how this is working in this slide, where we look at the membrane potential. Remember, the membrane potential is the charge across the membrane based on where the sodium, potassium, chloride, and so forth are located. So we have right now negative 70 millivolts. The inside is, and that's our resting membrane potential. Something has triggered a nerve impulse. The inside begins to depolarize. And if you look at my cursor, once it reaches that negative 55 millivolts, we are now in an all or nothing event that opens up activation gates on sodium channels. Remember the activation gate? Just a gate that opens up because of it's reached negative 55 millivolts. And then that allows more sodium to enter. The inside becomes more positive. That's what's shown here in the membrane potential. The membrane potential becomes higher because of the action potential taking place. And you can see the sodium is following the action potential. And once it reaches this peak of around 30 to 50 millivolts, the sodium inactivation gate comes in and doesn't allow any more sodium to enter. The potassium channel is open, and that's what's shown in this purple here. Potassium is leaving, making the inside more negative. So we're watching the inside become more negative. And we'll even have a dip that goes down below. And what is that dip called again when it goes below resting membrane? Hyperpolarization. So when it's going up, we have depolarization. So resting membrane potential, depolarization makes the inside of the neuron in that little spot more positive. Then the sodium channels close. Sodium isn't coming in anymore. Potassium channels open. That's what's shown here. Potassium leaves. The inside repolarizes. And then we actually have a dip where we call it hyperpolarization. During this phase here, the relative refractory period, you cannot have a second action potential occur because the potassium is leaving. Not until everything is reset can you have another action potential travel down a nerve. This is one of the reasons why we say a nerve is an action potential or is an all or nothing event. It reaches this threshold and that's it. And then it goes back into this refractory period. No more action potentials until everything's back to normal. And and that's what's shown in this picture here. You cannot, during this phase right here, you cannot have an action potential. So if you look at slide 4, you cannot have another action potential at this moment. That's the refractory period. It's only when you're back to normal and you're back to your resting membrane that you're able to conduct another action potential. So essentially, this is what looks like up and down this axon. You got these sodium-potassium channels. Now, what's not shown in this picture is the sodium-potassium pump is always in the background. But the sodium-potassium pump is just kind of what set everything up. It's not crucial for the next action potential to occur. In fact, you could probably have several, up to 1,000 action potentials, I think is what I remember reading, before the sodium-potassium pump would really become critical again to getting everything back to normal. Because again, it's just a little bit of ions moving in and out of the cell that causes these changes in millivolts. Okay, so let's look at how the conduction of an action potential, and then we'll look at an animation. But essentially, and again, we're going to get into a little bit more details about graded potentials and action potentials and how it's all triggered. For now, just realize that most of this is happening right at the beginning of the axon. And again, I'll explain how that nerve impulse started down the cell body. But for now, let's just focus on the axon. What happens in the axon that's not shown in this picture? There's sodium channels. So we're zooming in with the magnifying glass. We see sodium channels here. The sodium channels open, sodium enters into the neuron, making it more positive. And what happens to the sodium channels down here? They're going to open because of the positively charged sodium there, sending the action potential down the axon. So here we get some sodium channels opening. Sodium comes in, then these sodium channels are going to open and so forth all the way down the axon. Again, this is taking place within milliseconds. So you can see the diffusion of the sodium across the membrane is causing these other sodium channels to open because they're voltage gated channels. And so the activation gate opens. And then potassium leaves. And if you remember, the inactivation gate is in there closing the sodium channels. And so the only sodium channels that can open are in this inactive region. So the sodium continues down the axon, activating these other sodium channels. And so that's what we're seeing here. That's in the wrong direction. Okay. Let's look at this slide, and then let's look for an animation. And then we'll finish up this lecture with an animation in this slide. So what we have up here at the top of the picture is the dendrites. Again, that's that are found on the cell body. Receive information. Obviously, that's a nucleus, but that's not relevant to this discussion. These neurons are coming along and activating depolarization of the cell body. And how does it do so? Neurotransmitters like acetylcholine will open up sodium channels initially in the cell body and in the dendrites. And if the charge is strong enough, meaning it's over negative 55 millivolts, these sodium channels will open up in what we call the axon hillock. It's this part of the axon but it's part of the axon that's narrowing from the cell body. So we go from the cell body down to the axon, it slims down. Well, that part that's slimming down is called the axon hillock. And it's just loaded with sodium channels, just tons of sodium channels are present there. And this is the region that will propagate the action potential down the rest of the axon. And so what you're looking down, so we're going to look at right here now. But down here is pictures showing you this region. So just realize we're going to kind of looking at before and after. We got time 0. Looking at my cursor, we got time 1, time 2. And you notice we have little electrodes in, so we can measure the differences in charge. So we've had a excitatory response, meaning that we're going to trigger depolarization. Sodium channels have opened in the axon hillock. And in doing so, those sodium channels are going to make a sodium electrical spark that's positive, a positive spark in a sense, due to some sodium channels opening. The activation gate opens. Sodium enters, the inside becomes more positive. And as the sodium positive charge begins to diffuse and travel down the axon, it's going to trigger these other sodium channels down the axon. So it'll, if you're looking at my cursor, it'll trigger them here and start triggering them all the way down the axon, down to the axon terminals. And then there it'll be triggering the release of acetylcholine, the neurotransmitter. And so as the sodium is flowing in and the inside becomes more positive, the sodium travels down and will trigger these sodium channels to open. It'll trigger the activation gate to open. So the activation gate opens. See that? More sodium flows in. So now this is, before and after, time 0. So now these sodium channels will open. And what do you see happen to these sodium channels? Well, the inactivation gates come along and close them. And the potassium channels have opened. So that means this part, point A, is now negative again. It's undergoing repolarization. So the yellow, right here, we go back up. This is the before. Depolarization has occurred there. It's become more positive. Then this region number 2 becomes more positive down here, while the region right behind it becomes more negative. That's repolarization. Potassium is leaving the neuron. And so now the charge is traveling down the neuron. And so at point B, it becomes more positive. And then if you look at time course 2, point C becomes more positive. And point B becomes more negative, repolarization. So the action potential travels down the axon. So this region becomes positive, then this region becomes positive, then this region becomes positive. And as this region is becoming positive, this region is becoming negative. And as this region is becoming positive, this part is becoming negative. Again, repolarization, hyperpolarization, and then we're back to resting membrane potential, ready to go again. And that's the cycle that'll travel all the way down the axon. So let's watch a video to help provide the animation to this depolarization and how an action potential works. And again, this is a good place to look at some YouTube videos. I'll show you a short one, but it really behooves you to go watch a few more of them after this lecture. So let's take a look at an animation and then wrap up this lecture.
A nerve impulse is an electrical current that travels along dendrites or axons due to ions moving through voltage-gated channels in the neuron's plasma membrane. Voltage-gated channels open and close in response to an electrical voltage, so they are affected by changes in electrical charge around them. When a neuron is at rest, a charge difference is maintained between the inside and outside of the cell. This charge difference is produced and maintained largely by active transport using sodium-potassium pumps. The pumps send sodium ions out of the cell and bring potassium ions in. While other channels allow some flow of potassium ions back out of the cell, the sodium ions cannot easily get back in to replace the lost positive charges.
So obviously this is our resting membrane, and there's going to be negatively charged ions and negatively charged proteins and so forth inside it. But the sodium-potassium pump is the big part of maintaining this, along with some potassium channels being leaky.
The overall result is that the exterior of the cell has a net positive charge and the interior has a net negative charge. The difference in charge between the interior and exterior of the cell is called the resting membrane potential. A nerve impulse begins when a stimulus disturbs the plasma membrane on a dendrite, causing sodium channels to open. Sodium ions.
So this would be our activation gate. I don't see the inactivation gate on this particular diagram. But remember, neurotransmitters and various things are going to stimulate these sodium channels to initially open. And from there, it's just going to be the voltage that's going to change and open up the activation gate, as I described already.
Flow into the cell, lessening the charge difference at that location. If the change is enough, it will cause nearby voltage-gated sodium channels to open. This allows so many sodium ions to flood into the cell at that location that the membrane there is depolarized, with the local region.
So you can see how the next sodium channel opens because of this depolarization and this sodium channel making this region more positive.
Inside the cell having a net positive charge and the outside of the cell having a net negative charge. This affects neighboring voltage-gated sodium channels, which then open, moving the depolarization along the membrane. This moving depolarization is called an action potential. Changes occur behind the action potential to restore the resting membrane potential. The voltage-gated sodium channels close and voltage-gated potassium channels open. This allows a rapid flow of potassium ions out of the cell, repolarizing the membrane so that the inside is again negative and the outside positive. This is followed by use of sodium-potassium pumps to fully restore the resting membrane potential and to reestablish proper concentrations of sodium and potassium ions inside and outside the cell.
So you can see this region is becoming more positive and this region is becoming more negative. When the potassium leaves, this becomes more negative, relatively speaking. And then, of course, the sodium channel, sodium-potassium pump's always working in the background, but it's not immediately important for this part. Let's look at another one.
In the last module, we took first steps towards understanding the electrical properties of individual neurons. We learned how electrical forces and diffusion give rise to membrane potentials, and we learned how cells can generate and propagate signals called action potentials, or spikes, along the membrane.
So this is supposed to be the myelin sheath, and what this is going, I thought this was going to talk about the sodium-potassium pump. Let's pause this for a moment. Actually, we'll just watch a little bit of this video. I think it provides a little bit of kind of piecing the previous lecture and this lecture together. Here you can kind of see a simple reflex arc where we have sensory nerves picking up on a signal that sends it to the central nervous system back down to the effector. And then we'll kind of wrap it up from this video.
Respond to environmental stimuli using specialized cells called neurons. A stimulus is detected by sensory receptors, and the body responds through motor effectors. These cells working together allow you to respond very quickly to threats. When you touch something hot, heat receptors of a sensory neuron detect the stimuli.
You guys remember what the name of this neuron is? pseudo-unipolar. And then, of course, you can see the glial cells, like we have the Schwann cells here. And then, of course, we have our interneuron, which is found in the central nervous system. And then we have our motor neuron, that's going to be the effector neuron. So obviously, this little girl's playing unsupervised in the kitchen, but their mom or dad should be there. I'll blame her dad this time. Or actually, I'll blame both parents. Where are they? So anyway, she's cooking by herself, burns herself. The signal travels down the sensory neuron to the interneuron, and that yellow is representing the action potential as it travels down the neuron. So nerves can be affected and turned on in regards to the stimuli too, or the stretching. Either way, it becomes more positively charged, opening up sodium channels.
And send the information of heat to an interneuron in your central nervous system.
And that's deep.
From there, a motor neuron
travels through the interneuron and then back down through the effector.
Neuron sends a response from your central nervous system to the skeletal muscles in your arm, causing them to contract and pull your hand away.
And this can happen rapidly, right? Before you even notice it, you touch something hot and you're already pulled back before you really processed that you're burning yourself. So it's nearly unconscious control, subconscious, And so it starts to make you start to realize perception a little bit. I think one of the things about this class is you start to realize that your body's just interpreting the environment. It's almost like a computer, right? I mean, it's an action potential is like a positive stimuli, O's and ones and stuff like that. So here we have these nerve impulses traveling down. And then what is this perception is all up here ultimately. In fact, if this girl had spinal cord injury and the pain signal couldn't send to her brain, she wouldn't even know she was burning herself. She wouldn't be able to perceive heat. So it makes you start to realize, what are we really? I mean, what is real? What isn't? I mean, you started talking about the matrix a little bit. It's all up here. And it's all due to the sodium potassium pumps and channels and nerve impulses. It really makes you start to realize reality. What is reality? I don't know. But either way, I'm enjoying the ride of being alive and experiencing it.
The fundamental process of neural transmission that underlies this action occurs in all neurons of the body. Neurons transmit this information through changes in the electrical potential of the membrane by the movement of ions.
So obviously we have our sodium potassium pump making the membrane potential. Here's our phospholipids in yellow. We got our sodium channels, I guess, in green, and our potassium channels in purple.
Across the membrane.
That is all the watery stuff inside the cell. And then we have our extracellular fluid.
An electrochemical gradient governs the movement of these ions, resulting in an electrical impulse. The resting membrane potential in a neuron, when the cell is not firing an impulse, is established by the unequal distribution of sodium ions outside of the cell and potassium ions inside the cell, making the outside of the cell more positively charged compared to the inside. The electrochemical gradient is established and maintained by an enzyme called sodium-potassium ATPase.
So this is a sodium-potassium pump, and remember ATPase, excuse me, the ATPase breaks down ATP to ADP. So where else have you seen ATPase? The myosin heads, right? And that was in muscles.
When a neuron is stimulated, sodium ion channels open and sodium ions flow into the cell. This leads to a change in the electrical potential across the membrane called depolarization.
So the sodium flowing in causes depolarization. It might be initially started in the cell body, but the axon hillock is the trigger zone. where the action potential will either happen or not, and then travel down the rest of the axon. And that's the depolarization. We call it, it's actually the graded potential when it's in the cell body. And that will be described in a future lecture.
Polarizing electrical potential travels down the dendrites and over the cell body. Multiple electrical potentials will combine at the axon hillock.
So when these charges come together, if they reach that threshold of around -50 or -55 millivolts, that will trigger the action potential to travel down the rest of the neuron.
In a process called summation.
Summation just means it's adding up. And if it adds up to be -55 millivolts, the rest of the voltage gated channels will open, and then we'll have an action potential, which is an all or nothing event at that point, to travel down the rest of the axon.
If the depolarization is large enough, an action potential is triggered. Action potentials are all or none electrical impulses that maintain their amplitude and
So remember, that means sodium channels are opening right here, and potassium channels are opening here. Sodium channels are closing here. So this is becoming more negative, this is becoming positive, and then these are about to become positive. And as you can see, the action potential travels down the axon. It really doesn't travel up the other direction as a general rule.
strength down the length of the axon. The action potential travels down the axon when the depolarization of an area of membrane causes adjacent voltage-gated sodium ion channels to open. The influx of sodium ions results in membrane depolarization along the membrane. After a short delay, potassium ion channels open and potassium ions flow out, repolarizing the membrane. For the neuron to fire again.
Notice what was not said. There's an activation gate here and an inactivation gate down here for the sodium channel. So the inactivation gate is actually what's closed right now. That's not shown in this animation. And then the potassium is leaving.
The resting membrane potential needs to be reestablished.
And so then we have a dip where we have the hyperpolarization. I don't know if she'll mention that, but
Sodium-potassium ATPase is used to move sodium and potassium ions against their concentration gradients.
this isn't that critical. I mean, this is important for setting up the resting membrane potential, but it's not what is needed to immediately go into another action potential. There's enough sodium outside already to have another action potential travel down the axon. So again, this is just readily working in the background all the time.
Reestablishing the resting membrane potential.
Not critical by itself for that.
As the action potential moves down the axon, ions are diffused.
So did you see how that happened? We have an action potential traveling down, opens up the activation gate, sodium's going to enter.
Potential moves.
And then the next set of sodium channels are going to open because of the positive charge coming down the axon.
Down.
Now this one's opening, and then this one will open, and this one's going to close, and this potassium channel is going to open, and potassium's going to flow out.
The axon, ions are diffusing only a short distance, allowing the signal to move quickly.
And when that potassium channels are open, we have a refractory period also where we can't have an action potential yet. So here's the action potential traveling down the axon. It's going to reach
At the
axon terminals, and then we're at our synapse, where it's going to, and our synaptic cleft is the space, and they're going to release neurotransmitters that could excite this cell to have an action potential or excite muscles to contract, as we saw in the first lecture set. And we call that excitation contraction coupling. Remember how the excitation is the depolarization that I talked about, where sodium comes in the cell membrane of the muscle and goes down the T tubules and all that kind of stuff.
axon terminal, the electrical impulse passes to another cell at a cellular connection called a synapse.
We'll talk more about what happens at the synapse and how the neurotransmitters are released in the upcoming lectures. So what I would like for you to do, again, let me share the screen, is I would like for you to write a short, well, not a short, because this is actually a pretty long lecture, half a page minimal to 3/4 of a page of single space describing what you learned in this lecture. I would like for you to draw this slide, which is slide 45. And I would also like you to draw slide 42. I will make this worth 2 lecture points. Or excuse me, I'll make this worth, most lecture summaries are worth 2 points. I want to make this worth 4 points because of a little bit extra work. It's a pretty long lecture. It might be equivalent to 2 lectures. I'll look at the time frame, but either way, I'm going to treat it like 2 lectures. So this will be worth 4 points. Draw this, label and diagram it. Draw this, label and diagram it. What I recommend is looking at it, studying it, flipping it over, drawing. If you need to flip it back over and look again, go ahead. Just try to put it into your memory. You may be thinking, well, this is so simple. Sure it is. But I want you 3 months from now to be able to still have this in your brain. You look at it once and put it down, what happens? You forget it pretty quick. But what happens if I make you replicate it and repeat it a few times? Then it becomes more permanently implanted into the long-term memory. It's like anything in life. If you're trying to become good at music, good at playing an instrument, good at learning jiu-jitsu, dance moves, learning lines for a play, you practice. And I want you to know this, because how can you call yourself a biologist if you don't have a basic idea of how a nerve impulse works? Now, there's a lot of areas in biology you're not going to know that are specialized areas, but this is a kind of a general topic that a typical biologist should be aware of to call themselves a biologist. And so that's why I want you to really learn this how nerve impulse works. I think every biologist should have a basic idea of how a nerve impulse works to call themselves a biologist, even if they're an ecologist. You should have some idea, because you might want to get into behavioral ecology. You need to know how some of the machinery is working, even if you're studying flying squirrels. Just like a physiologist should have some idea of ecology. So, write a half a page, a 3/4 page, single space summary of what you learned. If you happen to make a double space, make sure it's over a page. And then I want you to draw this picture, slide 45 and slide 42. Explain what's going on. That doesn't necessarily mean you have to draw every single frame, but you need to be able to draw enough that I know that you know what's going on with a nice little summary. Be sure to diagram it. If you have an inactivation gate, you should be diagramming that. I also want you to look at some videos. Take some time to really learn this topic. Read. If you have a textbook, read it. If you don't have a textbook, read it up on the internet. Watch some videos. There's always some details that are missing when it's just something I verbally describe. You'll learn a few details, quite a bit more details, if you read a physiology textbook or a general biology textbook that describes this. And it'll help you put it into memory. Anyway, I hope you got something from this lecture. I will be posting it on YouTube soon, and I will be sending you this via email as well. Anyway, have a nice weekend and take care.